Hi, my name is Naomi Joseph, and I'm going to be giving a review of the paper written by Shukla et al. From Case Western Reserve University, and the paper is called "The Increased Tumor Homing and Tissue Penetration of the Filamentous Plant Viral Nanoparticle Potato-Virus X." Nanoparticles have proven their capabilities and strengths in previous research, showing they can carry drug payloads and deliver them directly to specific target sites. The chemical and physical characteristics of these nanoparticles really define its potential uses and can significantly impact a nanoparticle's fate. However, synthetically altering such characteristics to obtain a high aspect ratio can be difficult due to polydispersity and poorly controlled chemistry. Thus, it is more efficient to use nanoparticles derived from nature, such as viral nanoparticles or VNPs, with a filamentous material property giving them a high aspect ratio, superior pharmacokinetics, and tumor homing properties. This was the undertaking of Shukla et al. In this paper. The benefit of using VNPs is they do not cause an immune inflammatory response because they are not pathogenic in mammals. Furthermore, their structure is easily modifiable with the help of targeting ligands, drugs, and/or imaging molecules. And while most VNPs utilized in research and nanomedical applications are spherical nanoparticles, few VNPs with high aspect ratios have been investigated. The purpose of this paper was to investigate the filamentous plant VNP Potato-Virus X, or PVX, and compare its biodistribution and homing properties to those of cowpea mosaic virus CPMV and an icosahedral nanoparticle. CPMV particles and PVX filaments were purified from their respective plants. In Figure A on this slide, the PVX and CPMV modifications are shown, with both molecules being conjugated to an A647 and PEG5000 via lysine side chains. To ensure that these nanoparticles were formed successfully, they were analyzed via a transmission electron microscopy, or TEM, and these images are shown in Figure B. They were also evaluated using size exclusion chromatography. The reason the VNPs underwent pegylation is because this reduced undesirable reactions, prolonged circulation in plasma, and increased stability of the particles. Figure C shows the results of gel electrophoresis of protein subunits to confirm that both A647 and PEG5000 were covalently attached properly. UV visible spectrum was utilized to calculate the number of diamoieties per VNP. Finally, in Figure D, we see the zeta potential of both CPMV and PVX, and this indicates the degree of electrostatic repulsion between adjacent similarly charged particles in a solution or dispersion. Zeta potentials are important to consider here because surface charge is just as strong of a contributing factor when it comes to drug delivery and material characteristics as is that particle's geometry. One thing to note that although the isoelectric point of both CPMV and PVX are quite similar, their zeta potentials are opposites. CPMV has a negative zeta potential, while PVX has a positive zeta potential. The following results and their differences are the consequences of the physical and chemical characteristics of PVX and CPMV nanoparticles. Evaluation of tumor homing and tissue penetration properties of CPMV and PVX were conducted by intravenously administering the two VNPs into human fibrosarcoma and squamous carcinoma tumor models. The distribution and aftermath of the CPMV and PVX within the tumors was viewed via intravital microscopy. The two VNPs were dyed with different fluorophores to allow for direct comparison between images as shown in the figures on this slide. In Figure A, we see the overall tumor uptake of both CPMV and PVX. CPMV was dyed green, while PVX was dyed red. Taking a closer look, we see the distribution of VNPs differ mostly in the center of the tumor. Thus, in Figure B, when examining the intratumoral localization, there was a significant difference between the quantity of PVX in the tumor core versus CPMV in the same region. The enhanced permeability and retention, or EPR effect, of a nanoparticle, which describes the nanoparticle's ability to deliver cargo to a specific target, is determined by how well the tumor is vascularized and the reduction of lymphatic leakage near the tumor. The EPR of elongated nanoparticles nanomaterials such as PVX is more enhanced than for spherical particles such as CPMV. In Figure C, a confocal microscope was used to examine small sections within the tumor, and the results show that PVX has a greater distribution throughout the tumor than CPMV and can be seen in areas that CPMV was unable to reach as well. The next set of experiments described in this paper involved utilizing a mouse model of colon cancer with human xenografts. The purpose of these experiments was to examine the different VNP's ability to target and identify tumor homing, or cells from distant locations traveling to a tumor location. In Figure A, PVX and CPMV were injected into the tumorous mouse, and the organs where the VNP was injected and found were imaged under white light and fluorescence light. It is clear that the PVX VPNs showed enhanced tumor homing compared to CPMV. Figure B, the left picture shows two tumors extracted from 3 different mouse models, imaged again under fluorescence, and the corresponding quantitative data of intensity is shown on the right. Here we see that PVX had a greater intensity compared to CPMV. Finally, in Figure C, intratumoral distribution of PVX and CPMV is shown, and it is evident that PVX has a greater distribution than CPMV. The reason behind these results is believed to reside in the physical characteristics of PVX having a filamentous rod shape, allowing it to migrate into the tumorous tissue better than the spherical-like CPMV's VNPs. The two intensity graphs shown here represent PVX and CPMV's distribution within the plasma time over a 60-minute duration. It would seem intuitive that a VNP having a high tumor accumulation would be the result of having an enhanced plasma circulation time. However, this does not seem to be the case according to the figure on this slide. CPMV was calculated to have a half-life of 20.8 minutes, whereas PVX has a half-life of 12.5. In other words, CPMV has a longer plasma circulation time compared to PVX because it is not phagocytosed as quickly. However, this contradicts previous studies which show that positively charged particles have a longer plasma circulation time, as well as the theory that a high aspect ratio would lead to a longer duration in the plasma. Furthermore, it does not seem that circulation time is correlated with tumor accumulation, and some other phenomenon is the cause of the higher levels of PVX in the tumor shown previously. The figure on the right shows accumulation and biodistribution of each VNP in each organ and tumor, and then also as a percentage of the total tissue's mass. These results match previous research which demonstrated that CPMV has an affinity for the liver while PVX has a strong affinity in the spleen. In conclusion, PVX was able to demonstrate enhanced tumor distribution and homing properties compared to CPMV VNP because of its high aspect ratio and filamentous composition. PVX's shape and surface charge give the VNP its advantage in nano applications over CPMV. A few directions this research could be further taken includes releasing drugs into specific tumor regions and also determining why the plasma circulation time of CPMV is greater than that of PVX. And here are my references, and thank you for listening.